So thanks everybody for joining today. It's a pleasure. Today we will talk briefly introduce our project ACROSS, which is focusing on investigating latitudinal gradients of species richness and body size spectrum in multiple trophic levels and across ecosystems. So here you can see our contact details for anything that you can reach us from these contacts information. So, okay. This is us. We are the co-PIs of the ACROSS project that we applied. We got this project early this year from SPECTRE. It's the Iberian Society of Ecology, and we it's for 2 years. It's for early researcher careers from this society. We are these co-PIs that we are leading now the collaboration, and we are leading the data analysis in this project with together with other collaborators. So moving to the project, as you may all know, like there are these consistent latitudinal gradients across different taxa, ranging from birds, mammals, reptiles, amphibians. So when we move from tropics to polar regions, that we tend to have to lower a number of species. So the species richness decreases when we move from these polar regions to the tropics. And however, there is this large variation in the species richness also in the same latitude. So we observe different species richness levels in same latitude. So why there is this unexplained variation in diversity patterns? Several researchers try to explain this unexplained variation using different approaches, tested different hypotheses, but growing evidence suggests that individual level processes such as individual differences in body sizes can explain these biodiversity patterns. Body size is a key trait, as you know, that it's highly related to the metabolism across different taxa, and it's also reflects the predator-prey interactions, especially in aquatic ecosystems, because predation is highly size dependent. It has also implications in different biological organization levels, from individual to populations to community and trophic structure. As you all know, that one of the ways to assess these individual size differences is size spectrum, that it indicates the relative contribution of small and large individuals in the community, and it also reflects ecological efficiency and food web structure. It has been also used as an ecological tool, as an universal indicator of ecological status, for example, as an indicator of environmental change, species invasions, resource exploitation. For example, if we have more impact in the ecosystems, then we tend to have steeper size spectrum as an indicator of quantitative measure of deviation of ecosystem from reference status. So in this project, one of the objectives that we have is to identify the role of body size spectrum in driving latitudinal gradients in species richness. So we expect the latitudinal size spectrum pattern will determine this unexplained variation of the classical species richness climate correlation. The other objective is to determine these latitudinal gradient biological patterns of size spectrum for animal communities. So we expect there would be a negative relationship between temperature and size spectrum slope. And we are additionally testing if marine ecosystems have flatter size spectrums than freshwater and terrestrial. because they have larger food chains and they can sustain high biomass in larger size classes. So for this, we adopted meta-analytic approach, where we surveyed literature using from only animal communities, using only empirical studies that have individual body size information and latitude longitude information. We search Web of Science using these combinations of search strings, and we searched all databases in research area of environmental sciences and ecology from 1990 to 2022. We selected 1990 because in order to ensure the data quality and to access to the data sets. So in this screening, we ended up having around 9,000 articles. And then after the first screening, where we only screened the abstract, we assessed these records for eligibility, and we had around 500 articles. So far, this is the work in progress. So we are in the still of second screening that we are reading the whole articles to extract the information on body size spectrum. But so far, we screened around 225 articles where we excluded 100 because they were from experimental studies, they had simulated data, and these other reasons. And from these 125, 79 are on hold because they had some missing data. So we need to contact the authors of the papers for more information about these papers. In the end, we ended up having 46 papers to extract data. And from these papers, we extracted this information about geographical coordinates, sampling year, month, ecosystem type, biological organization, if it is from community, or if this study includes more than one trophic level, so if it's from food web. We also extracted data on taxonomic group, species richness, size spectrum methods, and size spectrum parameters of slope, intercept, linearity. So far, we had around 1,600 size spectrum slopes and around 1,000 intercept and linearity data from these articles. So here in this map that you see the locations of these studies where we extracted from these 46 articles. As you can see, most of them are in North Europe and North America. That certainly be like studies from Africa, Australia, and South America. So if you have any collaborators, or if you know somebody that may have this kind of data, that we can have a talk later, that they can contact us. Additionally, we will extract data about human influence and temperature using some external data sources. So far, as you may also seen in the previous slide, that this data is coming mostly from North America and Europe, followed by Asia. And if we see the habitat type, that we can see that the lake constitute the most of the studies, followed by streams and coastal habitats. And we have a few studies from soil, from terrestrial ecosystems. And when we look at the biological organization, we see that they are mostly from community. Yeah, they have community data. A few studies have food webs. And when we check the trophic, the trophic groups, that we see that most of these data are from fish, followed by zooplankton, and then invertebrates. And there are a few studies or just using primary producers, bacteria. And when we see the type of the data they have, we see that most of the papers have biomass data, but like the difference is not so much. So it's there are also like studies using abundance, and these abundance, most of them are measured as densities. And here we checked if the data is binned or not, but most of the data are binned, and they are also normalized and log transformed. So actually, this is the this is where we are at this moment. So we are still in the screening process, that we are extracting data from the other half of the data. We are also harmonizing the vocabulary that we use in the meta-analysis approach. And for today, we have identified some discussion points that we would like to discuss with you. That first one is about dealing with the confidence intervals and the effect sizes for model comparisons among these studies. And the other one is about collecting these climatic and human impact data for different ecosystems. And we also would like to discuss the type of modeling approach. But if you have other things to discuss, that we can, we can discuss later. So thanks for your attention. So this is it.